Place identical twins side by side. Their DNA is nearly an exact match. Their hands developed in the same womb, under almost identical conditions. Yet look closely at these two index fingerprints, and the paths separate. A line forks on one finger while continuing straight on the other. Even genetically identical people do not carry identical prints. If DNA does not dictate the precise placement of these lines, something else during fetal development must be translating microscopic variations into a permanent, unique layout. A fingerprint represents the physical record of an active building process, rather than a barcode stamped from a genetic mold. That process begins between gestational weeks 9 and 15. Each fetal fingertip carries a rounded swelling called a volar pad, providing the spatial canvas for the coming pattern. Around week 13, the first primary ridges begin forming deep within the skin layers. These early structures organize into the broad categories we recognize as arches, loops, or whorls. As the fetal finger grows, the existing ridges stretch out. The body does not insert new primary ridges to fill the expanding gaps. The adult print is simply an enlarged record of this early layout. Older hypotheses suggested the skin buckled under compression, or that blood vessels supplied a hidden template underneath. However, these mechanical ideas failed to explain what first divides uniform skin into perfectly spaced ridges. The true origin of the pattern rests on a microscopic chemical sequence that plays out before the physical skin ever begins to fold. In 2023, researchers discovered that a forming fingerprint ridge initially mimics the molecular machinery of an entirely different structure, a hair follicle. Early hair buds and primary fingerprint ridges both intensify chemical signals involving the WNT and EDAR pathways. They begin with the same developmental software. Then the paths diverge. A hair follicle recruits deeper cells to grow downward. The fingerprint ridge acts as an abbreviated subroutine, reusing the opening stages of the follicle building program but halting early. Before the epithelium visibly folds, EDAR signaling creates an invisible map of precisely spaced periodic bands across the tissue. Basal cells then translate that molecular map into shape. These cells grow rapidly beneath a stiffer upper canopy, forcing the expanding tissue downward to create a physical corrugation. While tissue mechanics build the final shape, invisible chemical signaling acts as the architect drawing the map. This leaves a central question. What forces uniform chemical signals to organize into perfectly spaced bands without a pre-drawn template? The answer returns to a mathematical idea proposed by Alan Turing in 1952. He considered how reacting substances spreading through a uniform field could self-organize. Turing's model uses two coupled influences, an activator, represented by A, and an inhibitor, represented by I. These reaction functions describe a contest. The activator promotes its own activity locally while simultaneously generating a wider-reaching inhibitor that suppresses that activity in neighboring areas. These final terms describe how each influence spreads through space. This balance of local reinforcement and longer-range suppression magnifies tiny, random fluctuations into regularly spaced peaks. In human tissue, WNT and EDAR function as the activating system, while BMP signaling serves as the primary inhibitor pushing back. Modern experiments involving perturbed embryonic mouse digits and lab-grown human skin organoids provided the biological proof. The results matched the mathematical predictions. When researchers reduced EDAR activity, the continuous parallel stripes broke down into isolated spots, a characteristic response of a Turing reaction diffusion system. Turing's equations describe the exact biological algorithm that governs localized stripe formation in human tissue. Shifting from the microscopic to the macroscopic, we can see how these parallel lines coordinate into complex, swirling patterns. Ridge formation does not occur across the entire finger at once. Instead, the process launches from three distinct initiation sites: the raised volar pad, the boundary near the nail, and the lower flexion crease. Three independent waves of ridges advance outward across the curved geometry of the fingertip. The timing of these waves determines how they intersect. This is combined with the specific domed or flat geometry of the individual finger. These collisions naturally produce the Y-shaped convergences that define arches, loops, and whorls. The broad category of a fingerprint is the chaotic geometric result of local chemical waves crashing into anatomical boundaries. Genetics dictates the proportions of the finger, the shape of the volar pad, and the signaling pathways, but it does not encode every individual ridgeline. While current simulations reproduce broad configurations, they cannot predict the exact position of every single ridge fork or sweat pore. This explains the differing prints of identical twins. Variations stem from minute differences in tissue growth rates, wave timing, and fluid dynamics within the womb. A fingerprint is a frozen, permanent record of a physical embryo building itself, a localized biological algorithm written into the skin.